Our first speaker this morning is Dr. Jonathan Lee, Senior Consultant in Gastroenterology and Hepatology at the National University Hospital, Singapore. He is also an assistant professor at the NUS Yong Loo Lin School of Medicine. He currently serves as the Director of Research for the Division of Gastroenterology and Hepatology. Dr. Lee will be presenting on his study on the microbiome in the development of HCC. He will be sharing data from the Elegance program. Dr. Lee, please.
Thank you, Prof. Chow, and thank you for the committee as well for inviting me for this. Without further ado, I'm Jonathan Lee. I'm from National University Hospital and the university as well. And today, I'm very happy to, I'm very pleased to present to you maybe a different kind of experiment and results, right? But this is from what you heard earlier, the success of the HCC. This is from one of the protocols as well. So there's many results from protocol 10, all right? But we're looking at the microbiome in its development of HCC, and most of the data I present today will be locally found throughout. These are my COI disclosures to the talk as well. So just to give a background set as well with microbiome, if you guys are not versed in this, and basically not just the bacteria itself, but it involves basically all the different living members, so archaea, fungi, viruses. These are all part of the living members, which are the microbiota. And then the products they produce, as well as the genetic material, as well as the cell wall components, this entire theater of activity becomes the microbiome as well. Why this is of interest to study is that our human genome project, which over 20 years ago, we realize with the human microbiome project that we have the functional capacity of about 10 to 100 times the amount of your human genomes. And unlike your human genomes, it's much easier to manipulate the microbiome productions as well. And in this case, it's all because the host and the microbes have some kind of synergy with some symbiotic relationship. When you can maintain that symbiotic relationship, you can maintain health. The gut-liver axis is a very good example of studying the microbiome, whereby the gut as well as the liver through the gut-liver axis has a bidirectional relationship. And a lot of this will be covered by my next speaker, all right, where he talks mainly about bile acids as well. But I'll give you an example. This is a very unique field, all right, where in the gut itself, you have the portal circulation. So all the metabolites from the gut, they will have to bypass, go through a portal circulation, through the portal vein, and the liver is the first conduit for all these molecules that go through as well. And then the liver will actually signal back. Mainly, we mainly talk about bile acids, but we are appreciating a lot more different molecules now that will come through. But as the next speaker will go through, bile acids can actually shape the liver and microbiome composition in the gut. And likewise, then vice versa, the gut bacteria that stays in the microbiome will then produce a second set of microbial metabolites that can shape immunity, or it can actually shape your metabolism as well. So we have to talk a little bit of what the known literature is now, so we actually know what to augment on. There's been quite a number of studies, all right, talking about development of HCC and its role in the microbiome. And most of the studies have come down to mainly three pathways that how the microbiome can actually contribute towards the development of HCC. The first, of course, is that it's a conduit and a mechanism to propagate chronic inflammation. If you have essentially bed bugs, and if the bed bugs itself produce a lot of these pro-inflammatory molecules that stimulate the host to actually result in a lot of inflammation, persistent inflammation will result in molecular circuits, and this can actually result in cellular damage and then resulting in tumor suppression genes or oncogenic genes resulting in eventual cancer as well. But as we now realize, a lot of it is regarding immune metabolism. And so the microbiome is actually one of the many avenues whereby a lot of the host enzymes will not be able to produce some of these metabolites, but through the microbiome, you can actually produce some of these metabolites, which also bypass into the liver, and by also have seeping out of the liver, so you also have all these other mechanisms that can result in cellular damage. And last but not least, as checkpoint inhibitors, we are getting more and more rampant in this use as well in the oncology space. We now appreciate the role of the immune system in the oncology pathogenesis as well as treatment. And the microbiome is one of the very key factors which actually try to regulate the tolerance as well as the expression of the host immune system. And this is very well manifested from a few studies showing that microbiome metabolites can actually regulate immune phenotypes in the liver as well. So where does the evidence stand? Evidence stand mainly comes from three different kinds of studies. One really is like Mendelian randomization analysis, and this basically takes cohorts where they have a lot of genetic variants, and they look at the genetic variants, which microbiomes would be like an environmental factor for a particular variant of risk to result in hepatocellular carcinoma. So they pick out about two main papers in this, and they identified certain the bacteria that are associated with environmental risk towards hepatocellular carcinoma if you have a certain genetic variant. You also have causality models in animal models to show this as well. So you can actually induce the bad bugs into animal models and then demonstrate the cancer after that. And then what is more important is you must show that it's reproducible, is validated in human studies. And this is where the observational studies will come in. And so there have been quite a number. What I can find is probably two or three human observation studies have been published. Numbers are a bit smaller, so there is and then the technologies of that time, they are very limited in terms of the depth of sequencing, but nonetheless, there was a clear signal in this sense to suggest that it's fairly important of the microbiome contributing to hepatocellular carcinoma as well. So I won't delve too much of this, but to give you an idea with the protocol 10, HCC 10 Elegance study as well. So this study actually followed up about 2,000 participants from the multiple recruiting sites. Patients all had some degree of risk towards developing liver cancer. And then they were followed up every 6 months. And then during the 6 months, it was suspicion leading towards the diagnosis of cancer. Then the endpoint of that cancer is reached. If not, then they will continue the surveillance every 6 months. So this microbiome data that I'm going to present for the next 15 minutes or so is that it's a nested study within protocol 10. We looked initially at the first 216 participants. We did this as a nested match for age, sex, and BMI. We have at baseline 216 patients as seen in the green and purple circles. Followed up for average of 5 years, right? 181 of them didn't develop liver cancer. 35 of them subsequently developed liver cancer, and we also serially monitored the microbiome changes in 10 of them. And then we wanted to look at signals of liver cancer, so we looked at another protocol, and we looked at matched hepatocellular case controls of 54 patients with hepatocellular carcinoma and their microbiomes as well. So the nested cohort patient characteristics is fairly messy, but I'll take you through the table as well. In the baseline samples as well of the 181 versus the ones that didn't develop cancer and the ones that subsequently developed cancer, because it's age, gender, as well as BMI match, there'll be no major differences in that. But you can see in terms of etiology and things like that, there are certain minor shifts which are covered in the next slide a bit more. But you can see the usual risk factors, the greater the stage of the disease. So things like lower albumin levels, higher bilirubin, certain inflammation of higher transaminitis. These are slightly higher in the patients that develop HCC. We took all these models, we took all these variables, we put it into a model to try and build the best model to predict, and without any surprise, alpha fetoprotein still maintains to be the main predictor, as you can see in the bar plot down there, and the only one after multivariate analysis. However, the cutoff for AFP still remains below the usual international cutoff. It's very small to see from the top here, but the cutoff itself is in the main bar down here will be around 10, which is the middle of the plot. Most of them don't even hit that cutoff of 10. And the AORC, if you were to use alpha fetoprotein with various cutoff, is about 70%. So the rest of the talk now will be really focused on microbiome and trying to convince you that there's a way to use microbiome to augment our current prediction for patients with at risk of hepatocellular carcinoma, to actually try and beat the AFP, as well as to detect patients that are non-AFP secretors as well. So the clinical factors that are associated with hepatocellular carcinoma and the development of which in this cohort is shown in this flow chart. So the main risk factors like alcohol, hepatitis, fatty liver, as well as cirrhosis, we had to counter for some of this as well. So you can see in the plots as well, surprisingly, patients that develop HCC from our nested study, there were fewer of them that had hepatitis B, all right, or C. There was slightly more of them that had fatty liver, and is significantly more of them had cirrhosis at the beginning as well. So from these 216 patients as well, we collected their stools samples, and the stool processing basically is we collected for this part of the study, we collected them in a protectant to protect their DNA because we're studying the DNA. We worked in this case with Emily, all right, to process the samples, and then we passed them through an updated version of the annotation pipeline as well. We sequence them fairly deep, all right? The usual practice in this kind of sequencing is to sequence them only at 10 million reads. We sequence them on average to 20 to about 30 million reads because we wanted a lot more depth. Because of the enhanced annotations as well, you can see that we annotated from this depth an average, a total of 1,800 different microbial species, all right? Even after passing the filtering thresholds of 10% itself, we are able to still do analysis on more than almost 500 different microbial species. And then we're looking at functional pathways annotated up to about 5,000 different CAC autology modules as well. So the first thing I would look at is basically in the microbiome space, you want to see something called beta diversity. You want to see whether the microbiome composition differs from a group to another group. So in this case, we are very interested at the baseline samples, whether the microbiome would be able to distinct the microbiome at the baseline for whether patients will subsequently develop HCC. And so what we do is we actually do a per maneuver test. In this case, we do 100 permutation tests, and this is the average of the 100 permutations. We find in this case certain risk factors that will predict whether the microbiome will disperse from one another. And down here, what you can see on the bar plot on the left is that the status in purple rank 3, right, whether they'll subsequently develop HCC is a significant result on whether they would subsequently develop different compositions as well. If you look at the dot plot on the right as well, it's a bit small because I want to enhance a couple of things. The purple dots are the patients that will subsequently develop HCC. The red dots are the ones that I told you earlier on that had HCC at the get-go already, just as comparisons. And the gray dots are patients that didn't develop HCC in the 5-year follow-up. What you can actually see is that all the purple dots tend to aggregate together and cluster together on one side. In this case, I'm trying to pull out the bar plots to show you in this PCA analysis itself is significantly shunted with increased directionality of this as well, that there are actually distinct signals and a distinct cluster of this significant beta diversity among patients. At the beginning, you can identify they have different microbiome compositions. So what about if we do a unsupervised approach? And doing an unsupervised approach, we identified there are actually 5 different clusters of microbes. So in these 5 clusters, if you remember the previous plot, this is a replication of the previous plot, but now we've been on not telling the machine which patients have HCC and which patients don't have HCC and which ones will develop HCC. They accurately bin out 5 different clusters as shown in the 5 different colors. And what I'm trying to show here is basically if you try to look at the clusters, the one that you remember before that's shunted towards the purple on the right, those are the ones that develop HCC after follow-up. And if you were to just filter them by etiology, whether they are more, to start off with, they're cirrhotic or whether they have fatty liver disease, what you first notice will be that you can actually try and see, all right, in this case, if I just bring out the pointer down here, if they had cirrhosis, this purple bar in the group 4 cluster itself is significantly higher, and this will probably represent this group as well, that their microbiomes are here if they're cirrhotic and they're more likely to develop HCC. If they have fatty liver as well, then they are more likely to develop HCC within the 3rd cluster. We are able to pull out the different microbes within the different clusters as well. So the cluster for cluster 4 will be here with associated cirrhosis, and the clusters here within for fatty liver. We pull out a couple of these examples. So E. coli, one of the very common bugs, belong to the cirrhosis cluster developing HCC. Similarly, Klebsiella pneumonia belongs to that cluster as well. And then for fatty liver, things like bibliophilia, which has been associated with a lot of lipid accumulation, increased inflammation, particularly in fatty liver, is mainly associated with that. So at the get-go, microbiome can determine composition differences, and the composition differences can be fractionated and stratified into basically etiologies and their subsequent risks of HCC. But what happens really? So if we were to look at patients, now we're looking at different index. We're looking at how rich the microbiome is within one sample itself, and we call this alpha diversity. If we look at patients that didn't develop HCC, subsequently developing HCC, and patients that already have HCC, you see this U-shaped diagram as shown in all these 4 different indices for alpha diversity in the box plots on the left. And what it's trying to show is that in the beginning, patients that are more likely to develop HCC have a decreased alpha diversity, but when they get HCC later on, somehow the alpha diversity goes up. What I'm hopefully convinced between the two is that it's not as that diversity is a good or bad thing, but what is actually happening is probably what I'm trying to show in this cartoon right here. In a normal microbiome system, in a normal patient, healthy patient, you get a very diverse microbiome. In this case of the HCC patients, there's something within the luminal environment. They have a decreased diversity as shown by the number and the richness of the microbial species. It allows niches to form, allows for increased selected populations or microbes to then form then after. And then when they actually get HCC, it's no longer the same set of microbes. They get the same richness, they get the same diversity, but you get a whole different pattern of microbe signatures that are associated with HCC. In this case, either passenger or driver mechanisms associated with HCC as well. In this, we're interested to find out what are the microbial signatures that were associated with patients at the beginning who were likely to develop HCC. We did this through both univariate and multivariate testing. To just cut it short, all right, we managed to identify through a series of methods. We identified 79 different microbial markers for patients at the beginning, at baseline, who were subsequently developed HCC. And we looked at the patients that developed HCC in the beginning, and we found 7 overlapped microbes as well. I'm just showing 2 examples down here. One example is a very high butyrate producing bug. In this case, was Bacteria homis itself. You see the signal consistent that at the get-go itself, the microbes are present, but if at baseline, the microbes are low, subsequently a HCC level is low, suggesting that the signal is there before the actual onset of HCC. This will be early signals for which. Similarly, another one which is more pathogenic, all right, in this case, a Streptococcus species as well. With this Streptococcus species, if it's at present at the get-go, even if they have cancer, the readout is a bit higher as well. We also start to look at the 79 microbes and whether they associate with the risk factors and the other environmental factors. And this I will go through a little bit at the end to tell you that even your microbes maybe this way, there are things you can actually try to do to probably change your microbiomes for that as well. Because we managed to sequence that deeply, we were also able to look at the microbial functions that were associated with this taxonomic changes. Because of the wealth of the data, we managed to get quite a number of differentially abundant microbes all beyond the FDR correction as well. So we identified over 600 different functional CAG ontology enzymes or proteins as well. And then if we try to bin them using enrichment analysis to see what is enriched and is as a combined process leading to subsequent HCC or protective against subsequent HCC, how to read this is any of the bubble plots to the right are associated with the development of HCC. Anything to the left of the diagram itself are more like protective or negatively associated with HCC development. And things that enrich, just to give a couple of examples, would be things like amino acid metabolism, in particular anabolism itself, because we have the directionality of the expression of this functional potentials. And these are usually now thought to be very important molecular metabolites that are involved in the immune evasion or the immune regulations of cancers as well. As mentioned before in the earlier slide, chronic inflammation is one of those very key components. If persistent for a while will result in persistent inflammation and subsequent cancers. And so in this case, bacterial cell wall biosynthesis, things like the peptidoglycans production and things like that will result in further inflammation and subsequent cancer risks. And things that are depleted are things that are quite well established in the literature, beneficial short-chain fatty acids as shown by one of the taxa I mentioned before, and things like the signal for bile acids in this case, because of the granularity, as our next speaker may go through many different bile acids as well, it's difficult to pinpoint the particular bile acids, but in our study itself, there's a diminished capacity to metabolize bile acids associated for which is cancer as well. If we take the 79 markers, we can develop a panel. And this is probably one of the highlights of the study because we want to try and see whether we can pick up HCC earlier or earlier miss in some of these patients as well. So using the 79 markers, we can derive this dysbiosis index. So in purple are the ones that develop HCC, in gray are the ones that didn't have HCC. You can see the score is much lower. You can see in the KM curve as well, the markers at present at baseline can predict up to almost 3 to 5 years when they subsequently would develop HCC. And if you were to modulate the score a little bit, I showed you earlier on the AORC for AFP is 81, but now for AFP is 70%, and down here 81%, this is significantly higher. We have started to try to simplify the model, and in this case, using a array of machine learning methods and things like that, we have tried to simplify the panel to a panel of 21 markers as well. And you can see the 21 marker plot against the 70 marker on the dot plot on the left. The performance is pretty much the same. On top of that, what is more interesting is the one on the far right. What we plotted is AFP, and against the score of the markers, the purple ones are detecting all the ones that develop HCC. What you can actually see down here is that a lot of these non-secretors can be picked up by the dysbiotic panel, either by the 79 panel or the 21 panel for subsequent risks. This is probably one of the last few slides, right? I want to just end the point that everything in this microbiome is potentially modular. Modular in the sense is you can manipulate the microbiome. And the most common thing, you are what you eat for the microbiome, right? Food, diet are probably one of the key aspects. So this dysbiosis index generally predicts quite well the HCC risk. We present two sets of data on here. On the left, we collected their dietary intake, and we looked at their dietary intake versus the dysbiosis index. The lower the dysbiosis index, the more likely they're going to get HCC. In this case, beneficial vitamins, nutritions, if they basically take more, the likelihood that the dysbiosis index will be low will be much lower. So in this case, a beneficial diet is actually more protective. And what we actually did is when we looked at, because we had interval follow-up of the patients, so we were able to bin the patients into whether, in this example, patients that took high PUFAs or they take low PUFAs. And what we found that taking low PUFAs generally, high PUFAs generally are supposed to be health beneficial for you, all right? So if you didn't take enough PUFAs and things like that, over time, your microbiome index for the risk get lower, and this, as you get lower, as shown in our score itself, you're more likely to be at risk of hepatocellular carcinoma. So this temporal data for that as well. So in summary, all right, we're quite excited, all right? Elegance represents basically a prospective cohort with very deep sequencing to actually demonstrate this distinct microbiome signatures for subsequent risk for HCC. Hopefully after this, there is convincing evidence that microbiome features can really differentiate at the get-go patients that are more likely at risk to develop HCC, and that is able, with more data coming in, to tease out the differential pathways, or whether if you're more likely to have fatty liver or you're more likely to have cirrhosis, the road towards HCC changes a little bit. But nonetheless, the signature using the larger panel, the smaller panel, works very well, particularly for AFP non-secretors. And then this is very modulatable by diet. And so more temporal studies to look at interventional studies in this case to see where you can intervene on those will be very important. So with that, I think it's really a concerted effort with the patient recruitment for this. And I thank you again for the opportunity to present some of this data, but we were happy to get questions at the end. Thanks.